Hi, everyone, and welcome to episode 776 of People Behind the Science. Today, we are resharing our interview with our guest, Dr. Rick Myers. Listeners, Rick does research in the areas of genetics and genomics. In particular, he's working to better understand how our genes and their expression contribute to things like our traits, health, and disease. He also examines how genes and gene expression can be regulated. And in this interview, you'll hear more about Rick's research and his life. So I hope you enjoy this episode of People Behind the Science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to People Behind the Science. Today, I am excited to introduce you all to Dr. Rick Myers. So, Rick, welcome to the show today. How are you?
I'm great. Thank you, Marie.
Well, we are excited to have you here with us today and excited to learn more about your life in science. But I do want to start by giving our listeners just a little bit of background information about you before we dive in. So listeners, Rick is the president, science director, and a faculty investigator at HudsonAlpha Institute for Biotechnology. In addition, Rick is an adjunct faculty member in the Department of Genetics at the University of Alabama at Birmingham. He received his bachelor's in biochemistry from the University of Alabama and his Ph.D. in biochemistry from the University of California, Berkeley. Afterwards, Rick conducted postdoctoral research at Harvard University, and he served on the faculty at the University of California, San Francisco, as well as at Stanford University, where he contributed substantially to the efforts to sequence the human genome as part of the Human Genome Project. Rick accepted his current position at HudsonAlpha in 2008. And now Rick has received many awards and honors throughout his career, including an Honorary Doctorate Degree from the University of Alabama, the Blount Initiative Award from the University of Alabama, the Pritzker Foundation Award from the University of Michigan, and also the Darden Lecture Award from the University of Alabama. Rick has also been named a Fellow of the American Association for the Advancement of Science. And listeners, Rick is here with us today to talk about his experiences in life and science. So Rick, we're really excited to get to know you both as a scientist, but also as a person. So can you start by telling us what you like to do with your time when you're not doing science?
Well, I guess spending time with my family. I have two adult children. One lives in Seattle, one lives in London. So that's a little hard because they're far apart, but that's probably the most important thing in the world to me. My hobbies are I like to cook, I like to garden, and I like to do woodworking. I've done quite a bit of woodworking. I still have all 10 fingers, so I guess I've been moderately successful at it.
That's awesome. So what do you like to build?
Furniture kinds of things. When I lived in California, we remodeled our house and I built an entire stairway, which was a major project and it's something that I hated leaving behind when I moved back to Alabama.
Gotcha. So have you built anything in your current house?
A little bit, mostly furniture and small things, but it's sort of a therapeutic thing for me more than anything.
Absolutely. I know I live in an old house now and we've remodeled now almost every room in the house and it's cathartic and you get that immediate satisfaction when something is done too.
Exactly.
Yeah. Well, wonderful to get a window into your life outside of science, but I do want to talk about your scientific side next. So Rick, can you tell us how you describe what you do at work to someone who might not at all be familiar with your field?
First of all, I guess I have two roles. I'm a scientist and lead a research laboratory, but also being the head of a research institute, I do lots of administrative types of things, strategic planning and tactical planning, administrative stuff, take care of faculty. We have 16 faculty here at my institute at HudsonAlpha Institute. So that takes a significant amount of time. I did similar kinds of things at Stanford. I was chairman of the Department of Genetics when I was there for quite a few years. So that sort of organizational part of science is really important to me. But I also keep my hand in, not in the laboratory itself. I haven't worked in the lab in many years. The field I work in is in human genetics and genomics, or I guess I should say genetics and genomics, mostly associated with humans. I really like trying to understand how our genes, those that we're born with, and the expression of those genes, meaning the readout of our genes, how they affect traits and conditions that we care about, things like diseases and disorders, really how our genes help make us healthy as well. There's a lot of basic science in there. And I'm very interested in how genes are regulated as well. And the reason I say genomics is because we do this not just on the detailed biochemical level, but on a broader whole genome scale.
Very cool. Well, I'm looking forward to hearing more about your projects as we go through the interview. And I think in talking a little bit about your work, you mentioned you're balancing organizational and administrative research responsibilities. I think it can be hard to get everything done in a day. So I do love talking to scientists about what motivates them. And Rick, do you have a particular favorite motivational quote or saying that really keeps you going?
I love quotes. I certainly love reading people who have done great things. So it's kind of hard to pick out one. I did pick out one, and it's because it's from someone that I have great admiration for, George Washington Carver. I suppose that may be partly because he ended up spending 50 years in Alabama as a major scientist at the Tuskegee Institute back in the late 1800s through the early part of the 20th century. And this man was amazing because of all the things that he did under extreme hardship. This was at a time when things were far worse than they are now in terms of the way we looked at any differences in individuals. So I admire him. He was brilliant and did great science. His writings were not extremely erudite, but they were deep. And I think there are two from him, but one I'll say is, since new developments are the products of a creative mind, we must therefore stimulate and encourage that type of mind in every way possible. And when you think back about how he was doing this in the Deep South at the time that he did this, he was African American, and how important that was and how he overcame some of the difficulties that he and others faced. I really believe in that quote. And what it really says is that we need to value education, but we need to value it for everybody.
Right.
The other one is from Albert Einstein. I should expect many people quote him, partly because it's easy to find them, but then he was so brilliant. But I really like the short one, imagination is more important than knowledge. And that will come in later when we talk about some of the things that I think are important. It's not just learning facts, it's what you do with them. And being creative and freeing yourself to be imaginative is, I think, probably one of the most important parts of being a scientist. But it's something that I think that everybody should pay heed to.
Absolutely. Now, you mentioned two of these monumental figures in history, George Washington Carver, Albert Einstein. I think these people are truly inspirational. And I think as a scientist, there are many people who might inspire you or serve as mentors or role models throughout your career. So, Rick, are there any others that really pop up in your mind as people who've been really instrumental for you?
I thought about that a lot because there are so many who played important roles in this. And really, I view them as role models and people that I admire. So I've had many. My two undergraduate professors who got me interested in science, because I didn't start out in college being interested in it, that guided me. And obviously my Ph.D. advisor and my postdoc advisor, those I think for most people may not be role models. They were for me in very different ways. But I would say during the time I was a graduate student, there was a famous scientist named Bruce Alberts. He's at UCSF. He ended up being one of my mentors when I arrived there as a young faculty member, but even in graduate school and before, I knew about his work. And what the inspiration or the role model for me is that this is a citizen scientist. Bruce actually taught me that we need to care more about which papers we're going to get into or which grants we're going to get or our careers or how we're recognized, but that we owe the world, and that means everybody, both service, our time, and our knowledge, just explaining to them what we do. So we have a big education outreach program here at HudsonAlpha where we spend lots of money, 15 full-time people, where we are teaching the public at 20 different levels, not just in classrooms. This is not just K through 12. It's everybody, politicians, healthcare providers, you name it. We're explaining to them what we do in everyday language. Bruce taught me that. He's a big role model. And he's also, by the way, a great scientist, was and continues to be.
Well, this sounds like a fantastic and really important initiative. I'm really glad you brought up some of this public outreach and citizen science efforts, because I think you're absolutely right. Teaching people what we do as scientists is so important. And I think it's really lacking in a lot of ways in our society today. Well, thank you so much for talking about some of these inspirational figures. And you mentioned something that piqued my interest. You said that you weren't really initially interested in science in college to begin with. So what really sparked your interest in science?
So both of my parents were educated, a pharmacist and a nurse. So I was exposed to that. I knew I was going to always go to college. I didn't really know what college was when I was young, but I knew I was going to do this. And I guess I had some interest in science because I grew up in the 50s and really in the 60s as a child and young teenager. And so the space program just was amazing. I mean, I collected every article and watched every minute of all the moon landings and the stuff before and after that. But I never really thought about being a scientist, because to me, a scientist was the stereotype from the movies from the 50s and 60s. And in fact, it seemed like something I wouldn't particularly be interested in, but also completely unreachable, because it seemed like you had to be both nerdy and a genius and completely in a different world. And then I learned later that it's nothing like that. I remember being told by someone when I was thinking about science, I guess by the time I got into college, and this was an older person who said, "Oh, but you're way too social for that," meaning the stereotype of scientists being in their laboratories or their ivory towers. And
right.
What I learned very quickly is science is probably the most social enterprise you could possibly be involved in. And part of the outreach stuff that I mentioned is related to that. But even on a day-to-day basis, where people were interacting with people, we're doing lots of things, and it's intense. And so all of that ends up being not like the stereotype. So what happened is I started college in 1972. I've been around for a while. Mm-hmm. I went to save the world from overpopulation, because this was at the beginning of the environmental movement. I'm still quite an environmentalist. I learned to modify my views a little bit with time, but I wanted to study demography, how populations work. And so I started to major in sociology, and I spent about 2.5 years doing that, but decided to minor in biology because I thought I needed it for that. And that meant I needed to take chemistry. And I had a chemistry professor for my first chemistry class in my junior year of college, my third year of college, who was a fantastic teacher, but then took me aside and said, "Would you like to work in my lab this summer?" And I thought he was crazy, because I'm a sociologist. Why would I work in a lab?
Right.
But I did. He was a physical chemist. We were studying gas reactions and had all this equipment. And within maybe 2 or 3 weeks, I learned why you do science. You're trying to answer questions. I had no idea what it was all about. You're trying to answer questions about the universe. This happened to be chemistry and atmospheric chemistry. And then you make discoveries, and then you report. I just sort of learned what the process was. And I said, I want to do this. So I ended up majoring in biochemistry. I worked in another laboratory. I spent a little extra time after college to get even more research experience. And then from there went to Berkeley, as you mentioned at the beginning, went there to graduate school, ended up working in a lab studying how proteins that affect transcription, they bind to DNA and affect whether genes get expressed, and also how they replicate. So I studied that, and then went to do my postdoc and had an interesting change in direction midway through my postdoc. I did my postdoc with Tom Maniotis at Harvard University, who's a wonderful advisor. And I was continuing to study how genes are expressed and how they're regulated, and developed a method for making pieces of DNA have mutations in them in the test tube as a part of that study. And that ended up leading to me developing a technology for detecting DNA sequence variants, or what we call mutations or polymorphisms, I guess. And Tom pointed me in the direction of human genetics and said, people in that field are really interested in being able to detect mutations because of the few things we knew about disease back then. So I started studying and approaching it that way. And Tom got invited to a meeting in Alta, Utah. I'm going to come to that a little later. That ended up inspiring me to change entirely into human genetics. So when I went to start my faculty position at UCSF, I started doing that. That led me to meet very early on a more senior colleague, David Cox, and he and I joined our labs together to try to find genes involved in diseases, developing methods for mapping our genomes, which led us to the Human Genome Project, and that's how I got involved in that. I was at UCSF for 7 years. Great position, great mentors and students, and et cetera. But moved to Stanford University, the Department of Genetics, where I was there for 16 years. My genome center, we were one of the first genome centers that the NIH funded. And as you mentioned, while we were also trying to find genes and disease and understand how they work, and we did, we were successful at that. We were also had a group that helped to sequence 3 of the human chromosomes, 11% of the amount there. So I was there for 16 years and then had an odd opportunity to come to Alabama. I'd had no intention or plans to do this earlier, but 2 gentlemen here, Jim Hudson and Lonnie McMillan, had wanted to start a nonprofit research and teaching institute that could be devoted to genetics and genomics. And I was helping them design it as I knew them from earlier days, and decided that I wanted to come run it. And so we came here And it's unusual, but it was a cotton field before we started. Now it's a campus of 150-something acres, 4 buildings, about 800 people here on the campus now and growing in Huntsville, Alabama, which is where NASA started. It's a big science city. So it's turned out to be both a wonderful opportunity, but to build something that is sort of like an academic enterprise, but has a lot less bureaucracy in many ways. We've recruited wonderful people to come here. We do a lot of work more collaboratively than you can sometimes in academia. So I'm here, and I'm here to stay.
Well, Rick, it's wonderful to hear more about your history and how you got to this place where you are today. And this institute sounds phenomenal. And it's great that you really got to see it form from the ground up. Like what you said, it was a cotton field before, and now it's grown into this bustling campus. So what is it like working at Hudson Alpha?
Again, I'm going to go back to the inspiration of Bruce Alberts. We push very hard team science and doing something in sort of an integrated way. Science is not just going in the laboratory and doing something, making discoveries. It's a social enterprise, and it's something that's really important to the world. I think basic science as well as applied science. And we're kind of a hybrid of that. The nonprofit side has 16 faculty. We have appointments at various universities. And because genome science is so complex and multidisciplinary, we never do anything on our own. No scientist really does for that. Mm-hmm. But we never do anything completely alone. Lots of collaborations. We built a large genomics infrastructure, which is not just a lot of the equipment, but it's the knowledge and understanding what kinds of questions you want to ask and how do you want to apply them. And then our field, this particular field, is a huge amount of data generation, but a huge amount of data analysis and handling. And I'm sure you hear this. Computation, data analysis, statistics is a bigger part of our efforts than actually generating the data in general. It's wonderful here also because we're not a university. We don't have a tenure track system. We don't have rewards based on proving that you did everything by yourself, which I think has always been a little bit of a fallacy in that part of academia. So instead of that, we encourage and reward for collaborative team science kinds of behaviors. And I think we've done that really well here. And that's what I'm proudest of. And it works for most people. Maybe not for everybody, but it works for most people. So we've been able to be very productive because of that. The other part is because we built this infrastructure, many people use our infrastructure through two of our faculty members, but one in particular, Dr. Sean Levy, who runs what we call the Genome Services Lab, which is often fee for service, but it ends up leading to large numbers of collaboration. So we actually have generally way more work than we can actually do, but right. We initiate a lot ourselves, and each of our laboratories have research programs, but again, a little different from the way it was at Stanford and Harvard and all the other places that I've been in, that, for instance, our labs are kind of meshed together. People sometimes say somebody works in the Myers lab here, but it's really not the Myers lab. It's my lab. It's Dr. Sarah Cooper's lab. It's Greg Cooper's lab. There's several other faculty that we're almost all one. Everybody gets recognition for everything, but it just makes it much more fun doing it that way. And it's actually more efficient. We get a lot more done by not being isolated, I think.
Absolutely. So can you tell us about one of the things that you are working on right now that you are so excited about there?
Okay, so no one ever accused me of being focused. I do a lot of different things. And again, partly it works because our labs work together. We collaborate with lots of people. I have been interested in diseases of the brain and how the brain works. I'm not a neuroscientist, but I've learned a lot from starting 30 years ago, working first on Huntington's disease and then other neurodegenerative diseases. And so I'm going to pick one project out of many that I'll just mention now. And it's an interesting idea. There are many neurodegenerative diseases are dominantly inherited. There's a single gene, and you really only need a mutation from one parent, and then you get the disease. And so the mutant gene expresses a mutant protein. It's not that it turns it off in general. Generally, you're making a mutant protein, like in Huntington's and the 3 Alzheimer's genes that are known, ALS and Parkinson's, almost all have some dominant genes. And it looks like the reason that you get disease, and it's often later onset because there's some accumulation of damage, the reason you get disease is because the mutant protein is like a poison. It's bad. If it wasn't made at all, it'd be good, because from the other copy, you have the good protein. Mm-hmm. So we started undertaking maybe 6 months ago a project, and it's burgeoned to several projects, to see if we can figure out how to turn off the mutant gene in a cell, but not the wild type gene, not the normal gene. So in other words, you're not just downregulating both genes. It turns out you need the normal protein for Huntington's, for the Alzheimer's mutations, for ALS genes, et cetera. You need the normal protein. So can we get rid of the mutant one only? We call that allele-specific gene knockdown or gene turn down. Let's just take the Huntington gene as one. It's a single gene on chromosome 4, accumulates a toxic protein from one copy from the mother or from the father. So we are trying to figure out every last little piece of DNA in and around that gene that controls its expression, that turns it on or regulates the levels of expression, as well as all the transcription factors and chromatin marks that are involved in regulating when and where it gets expressed. And so we're trying to find all of those elements, and we're making progress. It's based on stuff we do for other kinds of projects, understanding gene expression. We're now working on, can we use that knowledge to now turn off the gene from the mutated chromosome and not the wild type chromosome? I just think that's exciting because while that's all in cultured cells, it could end up leading to animals. And it's a little hard to think about how you turn off genes in living organisms, and especially in the brain because it's so complex. But never say never. I think there are ways that we can think about that as well. And of course, part of this is based on the fact that we now have new gene editing techniques, CRISPR-Cas9 and related things that are just changing the speed at which we can edit the genome. So that's part of that project as well.
Well, this is a phenomenal work, Rick. And I'm curious, if you find a mechanism, for example, for Huntington's disease, do you think that mechanism and the way that you would address it is disease specific, or is this something that once you figure it out, you can apply it to any disease?
That's a great question because the answer is apply it not to any disease, but perhaps to any dominantly inherited disease, meaning knock out just one or the other. You can certainly take recessive genes and maybe correct a mutation where a gene is missing. That's not what we're working on here. But the reason it's applicable is that there are 3 genes for rare forms of Alzheimer's that are single genes that are dominant, could do exactly the same thing. And in fact, we've already started doing that with Alzheimer's. And ALS, the same thing. We worked on ALS for quite a while, and some of the genes that are known for that are dominantly inherited in the same way. So it could be a general paradigm. And other people are trying to approach this in different ways. We're just approaching it from can we use deep knowledge of gene expression regulation and use that to help try to do something about something that's very harmful.
Well, this is a really exciting area of research, and it's amazing to think about some of the possibilities that might lie in our future following this research path. And I think we've talked about your career, though, in terms of the wonderful things that you've done, but we haven't yet touched on, Rick, some of the challenges and struggles and problems that you've faced. And I think every scientist goes through them. So I love to share these stories with our listeners so that they get a realistic and holistic picture of the life of a scientist. So do you have a story you can share with us today?
Let me preface it to say that, and it may be obvious, I'm a pretty optimistic person, and I certainly can have setbacks, but I think I bounce back from them just because I'm optimistic. I had a close friend when I was a postdoc who was the opposite, who got very upset with me because I was happy all the time, but he wasn't. But certainly you have setbacks. I mean, there are lots and lots of challenges. Actually, let me do a general one first, and then I'll give you a specific one. One of the general things is, especially what I have to do as a leader of an institution and being the head of this, is that there are always problems where there are conflicts and issues that arise, and they're big, and they take a lot of time. But that's true at every level. And we're people, so we're going to have conflicts. And so part of it there is, it's not that it's a burden, but that's just a part of everyday life. And I have to deal with it in a big way because of the responsibilities I have. So those are some of the kinds of setbacks, I guess. But I do find that if I have some big conflict, not with me, but with something I need to solve here, that sleeping on it sometimes almost makes it go away because sometimes they're not really conflicts. But let me give you one specific one. So we were one of the first genome centers when I was at UCSF, and then we moved it to Stanford, starting in 1990 when the Human Genome Project started. We were one of the first ones funded by the National Institutes of Health. It was great. It was exciting. It's so funny to look back on that because we moved at a snail's pace then compared to what we can do now. But that was the part of the process of learning how we were going to sequence one person's whole human genome. So our group at Stanford did pretty well. We then put in for the renewal for the next round of funding. I think it was a 5-year cycle. So at the 4th year, we put in a grant and we just barely didn't make it. So I thought not the world had come to an end, but I've got this genome center now. We're going to probably have to figure out how to close it down. And we were loving the work that we did. So it was sad. And also all these people's livelihoods, I should say, were involved in it. But right after that happened, because we were good at doing a couple of things, we had another funding agency, the Department of Energy, who was doing a significant part of the human genome, approached us and wanted to know if we wanted to work with them because they wanted to have us join their team. And so we did, and we ended up being very successful in that. I'm not bragging on myself. I'm bragging on my people. I had wonderful people who worked with me there who really helped to make a major impact on the first human genome sequence, which of course is historical. So the lesson I got from that is don't give up. Also, be willing to adapt. There was some creativity there. It was different. It was a different pathway. And by being willing to look at these broader opportunities rather than just licking our wounds, I think we were able to overcome it.
Definitely. And I think this open-mindedness in considering and looking for other opportunities is a key to success as a scientist, especially today.
Even more so today, absolutely.
Well, thank you so much for sharing one of these challenging moments and how you worked through it. But we don't just want to talk about the difficulties, Rick. I want to give you a chance next to talk about one of your amazing successes. So is there a story that you have of either a big win or a small victory that really meant a lot to you?
There are lots of them. I've been doing this for a long time, so there are lots of them. But let me use a recent one. And this is one that my laboratory, working closely with Sarah Cooper's laboratory here. She's one of the faculty members here. So well, we do lots of collaborative stuff together. And then also with clinical laboratories at University of Alabama in Birmingham. And this has to do with pancreatic cancer. So we don't see patients here for cancer or anything like that. We actually do have a small clinic here that diagnoses diseases in undiagnosed, especially kids, but adults as well. But we end up relying heavily on collaborations with physicians and physician scientists. And so this study was a nice medium-sized win. I'll call it that, not a small and not a big. Mm-hmm. So we were looking at the tumors from people with pancreatic cancer. My mother had pancreatic cancer and died from it, actually. This had some real personal feeling for me too as well. So we were looking, we had maybe a little bit more than 100 tumors that people in Birmingham, as well as several other medical schools around the country, that had removed and dissected. It's actually hard to get these because many people don't have their tumors removed with pancreatic cancer. It's usually too late when it's discovered, so they don't even do surgery. So we did these, and we threw our genomic tool chest at it, is one way to say it. We looked at various things about the genomes of these 100 and plus tumors and actually compared them to non-tumor as well. And what we learned was we found some genes that were present in cancer, but it wasn't anything new. By then, another group had published some. It was useful to know this. There's some genes that are clearly overexpressed in pancreatic cancer, but not in normal pancreas. And so it wasn't a loss to learn that. It was kind of seeing something that other people had seen. But we decided to look at other questions in the data set. And so one of the questions was not just cancer versus non-cancer, but something else different about the 100 individuals there. So most people die from pancreatic cancer, unfortunately, very, very quickly within a year or two, and sometimes faster than that. A smaller fraction live longer, and sometimes 5 years or longer. So it turns out our genomic data that we were looking at, we found signatures that distinguish people who live a long time with it versus those who don't. And that kind of surprises. So it was one of the lessons there, which we try to do all the time, is to look at a broad set of questions rather than just have one that you're going in. You obviously don't want it to be just a fishing expedition. So by asking the question, is there anything different about long-lived versus short-lived? And it was a huge genomic signature that was reproduced in public data that were available. So that was gratifying as scientists to learn something new. But the reason why it's a win for us, and we think it's important, is that you can use that information as soon as a patient is diagnosed with pancreatic cancer. You can determine which type they have. Is it going to be the fast moving or maybe slower moving, and then treat differently depending on which genomic signature you have. So indeed, our colleagues in Birmingham, we're working with them as well, but they are beginning a clinical trial where they use our genomic test to say which treatment should this patient versus that patient get. So I like that one. We learned some basic science, but it's also pretty quickly applicable.
Well, congratulations on this win, Rick. I'm going to go ahead and round this one up to a big win. You called it medium, but I think you're being modest here.
Well, we're excited about it. It's hard to do these. And one of the important things I meant to emphasize there. So we did this on a modest size number of people, 100. You need large numbers often in order to, and you can get fooled easily by just spurious results. So one of the really, really good things about the direction that science is going is that there's several large projects, the TCGA being the Cancer Genome Atlas being one of them, and the ICCGA being another, where large amounts of data, genomic and otherwise, are being obtained from, these are all about cancer in this case, and put into public databases. So we were able to replicate our results by looking at data that people had not analyzed as such, but they were there in a public repository. This is the way science needs to work in every arena, not just in genomics or cancer genomics. And that played a big role in this as well.
Definitely. And I think the story really showcases your optimism, maybe not getting so bummed out because someone had already published what was going to be the main findings of the study, or not really giving up on the project, but keeping open-minded, looking for those other opportunities or other questions that you can ask with the data set. So I think those are really good take-home messages for our listeners as well. So now we talked a little bit about your scientific side, Rick, but I do want to dive back into the world outside of science and talk about books for a moment. I love getting book recommendations from everyone we have on our program and sharing them with our listeners. So do you have a book that we can add to our collection today that you really enjoyed?
I like to read a lot. I try to alternate between fiction and nonfiction. And for the nonfiction, it's often science or science related, but it's also society related, how we think, how we do things. I think probably the one I would pick is just a recent one. I have several like this, called The Undoing Project. The subtitle is A Friendship That Changed Our Minds. And Michael Lewis is the author, and he wrote it about these two very famous, actually psychologists. They were from Israel, Daniel Kahneman and Amos Tversky. Tversky was actually at Stanford. I didn't know him, but he was at Stanford, and I overlapped with him when I was there. And basically, they really studied how we make decisions, how we decide on things. And Moneyball, the movie and book, that was basically about their approach, basically to use data and non-emotional ways of making decisions. You hire a new hitter for your baseball team because you think this is the way we've always done it. You think about these particular features, and they actually found that it was more successful to do this, not in a robotic fashion, but to use, they call it behavioral economics. So one reason why this had such a big impact on me, I know lots of people who knew more about these guys and their work than I had, but it was kind of my first introduction to them. But it's really a way of also using big data and mining. There's a lot of data out there. And if you can figure out how to get at it and then analyze it in ways, and I have to tell you, I am not one of these people like the young Turks in my group or all these young scientists that I'm around. They're the ones who are figuring out how to do machine learning and use these big data sets. And a lot of it is quantitative tools. And it's just remarkable that Pancreatic Cancer Project is a small version of that, but there are many others that we're seeing where we're sitting on a gold mine of information, and it's figuring out how to get at it and interpret it. So that book had a big impact on me. I really like it. It's made me read some other ones about them and by the two scientists that Michael Lewis wrote about.
Fantastic. Well, we will add this to our list online for our listeners to find there. And I'm glad you mentioned it. I actually hadn't heard of it. So this is new to my list as well. And now we've talked about some different aspects of your career as a scientist. And one thing we haven't yet mentioned really is some of the opportunities to travel. We talked about the different places you've gone for the different stages of your career training. But I think it's so important for scientists to travel, to go to meetings and meet other scientists and share their work. So do you have a favorite place, Rick, that you can tell us about that science has taken you?
Sure. Let me preface to say that one of the things I just absolutely fell in love about science and about doing research that I really didn't learn until graduate school is, I don't really want to call it opportunities to travel because it wasn't always me going elsewhere, but the international nature of it. I mean, even in the 70s when I was in graduate school, before the wall came down, I got to meet scientists from the Soviet Union or from East Germany or from places and from all over the world, really, because they'd come and give seminars. I would go to conferences. And that had a big impact on me about there really aren't and shouldn't be any boundaries. And going back to one of my heroes, Bruce Alberts, a major part of his whole life, and even currently, he visited us recently, even currently, is being ambassadors and doing science with people all over the world, including places that don't have a lot of resources. It's a universal language, and it's also, I think, and he thinks, and many do, a great way of bringing people together and actually leading to peace rather than always sparring with each other. So I've been to a lot of places, not every continent, but a lot of places in Asia and Europe, Canada, obviously. And I'm going to mention two of them. One of them that had a big impact on me was going to St. Petersburg, Russia in 1991, where we were trying to be emissaries of science. That just was amazing to me because I'd never seen anything like that. And it was also just wonderful to have scientists from all over Russia and the other countries to interact. But let me tell you one that had the biggest impact on me, and I mentioned it a little bit earlier, and it's in the United States. It was Alta, Utah. So it's at like 9,000 or 10,000 feet. I get altitude sickness very easily, and I do not ski. I grew up in a place where you don't not have friction under your feet.
Right.
We didn't have ice skating or skiing in Alabama when I was growing up. And so I've never been very good at it. I've tried it a little bit. So this is a big ski resort, and there was a meeting in 1984 that 19 people attended. It was in some resort there. I never once went out in the snow, but mostly suffered from altitude sickness the whole time I was there. I had a wonderful roommate there at that meeting, a guy named Maynard Olson, who is, again, another one of my heroes. Tom Antiotis sent me to that meeting because he was invited, didn't want to go. We got invited because we had started to publish these methods for detecting mutations, for measuring whether mutations were present. The Department of Energy held the meeting, and they were trying to figure out, is there a way we can measure the germline mutation rate? Meaning, what are we born with that our parents don't have, but we inherit from them, but we inherited from sperm or egg, but they don't have the mutation themselves? That would be the germline mutation rate. Turns out it's really, really low. They knew it was low. And one application of this is that the DOE was working with groups in Japan to follow, since World War II, since we dropped atomic bombs on Nagasaki and Hiroshima, to see whether the people who survived those bomb blasts passed on more mutations to their children than people who had not been exposed. And we were at this meeting. George Church and I were the two young scientists there. These were all old-timers who were using all sorts of methods. They were interested in my method because it actually measured mutations directly, but they had all sorts of cell biology methods. And the conclusion in the middle of the meeting was the mutation rate is so low, we'd probably have to sequence the human genome to figure it out. And everybody laughed.
Oh, wow.
By the way, I didn't say that. I didn't know what was going on.
Right.
One of the more prominent scientists there said that. So everybody laughed because it was so ridiculous. It was 1984.
Right.
The DOE folks went back, and a couple of years later, they said, we need to sequence the human genome. That's when the whole efforts with NIH and others and people in England and later other countries all got involved in saying, we need to do this. So that meeting had an impact on me in a big way because it made me want to be in that field, and that's affected what I ended up doing. But I think it affected the rest of the world too, because I think that maybe we would have come along to do the Genome Project anyway later, but I bet you it happened a lot faster than it would, under lots of controversy too at the time, because of it seemed like it was a different way of doing science. So that was a weird trip where I was miserable the whole time because I was ill and I was snowed in around some really smart and interesting people that it had an impact.
And you witnessed the birth of the Human Genome Initiative. That is amazing.
I'm not taking any credit. I just happened.
Yeah. You were an observer on the sides. I like it. So did you make it out and try and ski?
Nope. Never did.
Well, I would say I'm with you on that one. I grew up in Cleveland, Ohio, where there is actually quite a bit of snow. Not a lot of skiing, but I've never tried it myself, and I'm kind of afraid to be honest. My coordination is perhaps not up to par. So wonderful to chat about one of these exceptional travel opportunities. And I think you're absolutely right in talking about the international nature of science in general, and that it does bring people together from all different countries in one room and working towards a common goal, which I think is amazing.
There's a lot of evidence that that's absolutely the case, and I wish we could do it more and convince our legislators and our leaders how important that is.
Definitely. And I think throughout our conversation, we've sort of been talking about different aspects of science, such as the creativity involved and all these different elements that people may not think of when they think of science. There are these stereotypes out there, and we like to break them on our program so that people like younger version of you are interested in science and are maybe not turned off by that kind of career. So I love sharing the side of science that's more human, more funny, more fun. And I think there are so many scientists who are like this. So Rick, do you have a story of your own that you can share with us today?
Yeah, I thought of a lot of them, and some of them I can't say on a family-oriented show like this, but I learned this fairly early in my career. And one of the things I love about science is that, and maybe more academic science than in companies, but actually it happens in companies too. I mean, Apple Computer is a great example of it, where there's tolerance of wackiness, or at least another way of saying that, a tolerance for not being just like everybody else. So
right. Unconventional. Yeah.
Yes. So embracing some of that, because you get some creativity about that. Now, unfortunately, that also leads to some of the wackiness being really bad behaviors at times. And so it's not always a good thing. But I think that if we had a set of rules where this is the way you always behave and you don't ever step outside the box or color outside the lines, we wouldn't be moving as fast as we are. And I have numerous examples of people that I've known like that I am really glad we tolerated. Sometimes they're misanthropes, they don't like people. Sometimes they're just odd. And some of those have led to some remarkable successes. So I'm not going to do any of those by name, even though there's some striking ones. So then you had asked a little bit about, are scientists always serious and just hardworking all the time? And of course, the answer is no.
Right.
And there's no stereotype. They're all over the place here. So one of the other things is that what do you do in the culture of a particular laboratory? What do you do to unwind? So I'm going to use an old version of this. It's kind of trite, but I just remember this well. When I was in graduate school, I, along with another fellow, were the first graduate students for a young professor named Robert Chin, T-J-I-A-N at Berkeley. He's still at Berkeley. He ran HHMI for a while. And this laboratory, I swear we worked 100 hours a week.
Oh, my goodness.
I would come in early in the morning, work all day, go home for dinner. I lived close by, walk home, come back, and work until midnight. And frankly, it didn't seem like work. I loved, and most people in the lab loved what we did. Sometimes that could get overwhelming. But of course, if you're going to do that, you better enjoy yourself. So we had loud music in the lab, which we had to close the doors so we wouldn't bother the rest of the building. And then learned once the lab got larger, you had to be respectful for everybody. So you had to make sure everybody was okay with that. We had 2 English fellows in our lab, which then led us to put a dartboard up into one of our little rooms. And so we played, this is real darts.
Right, not the plastic tips, yeah.
I got to be very good at that. I can even beat my British comrades there. And then the lab did go once a week to a Chinese restaurant that was half a block away. Tej let us at least leave for that. He came to go to the restaurant with us. We ate family style around a big table. There were maybe 7 or 8 of us, and it eventually grew to 10 by the time I left there. And I swear it was like, I grew up in a big family, so it had some similarities to that, but we would sit down to eat. And if you weren't aggressive about getting something on your plate, you didn't get it. It was it was like a bunch of animals. Mostly guys, but there were women there too. So it just reminded me of growing up like that. But it was one of those things that was part of the lab culture. Now, I don't think anybody needs to work 100 hours a week in order to be successful. That was a different time. It was also fun along the way. But what I think is important is to have some perspective. So I learned especially when I started running a laboratory, that vacations are really important. Family is really important. There is no reason you can't have a very successful career, and even when you're younger, have children and be able to make all of this work. There's some challenge there. I have tried to make that a part of the culture of HudsonAlpha and of the department that I led when I was at Stanford within my lab for all the times that I've had it. So don't forget to be a human being in all this. And I think if you have that attitude about a place, then it's much more fun to be there.
Definitely. And I'm really glad you brought up these issues of balance, because it is something that a lot of young scientists worry about going into the career. And it sounds like you guys had a good mix where you really loved the work. So you worked really hard, but you're also having fun playing darts or having these group outings for meals where you just kind of leave your table manners behind and get in there. And I think that really is important in science and kind of building that lab family environment. And I think with your lab family, not only do you have fun in the lab and those sorts of things, but it's really great to talk about the big questions in your field together. So that's one thing I love bringing up on our program is the big question. If I gave you everything, what would you answer? So Rick, funding, staff, technology, feasibility, all of that aside, what is the one question you are dying to know the answer to?
It's a big question. And it really is the whole question of how our genes affect phenotypes, meaning disease, how we respond to the environment, et cetera. It's not all genes, it's gene and environment. And I think this is a project that should be done not by me, but by thousands of scientists around the world. Some places are trying to do this, and that is a Million People Project. A big epidemiological cohort of large numbers of people who want to participate. Could say it's a little bit like the Framingham Project, but with genomics and other things added to it, where these groups of people who want to participate, and this needs to be a broad swath of both genders, every ethnicity, various ages. It should be a longitudinal type of thing where information is gathered over the years and maybe biological materials like blood or saliva, various things. To do this kind of thing where you have this large cohort of individuals and, more important, very, very large number of bits of information about their phenotype, meaning clinical information, etc., that then are available to every scientist, anybody in the world, to then use that information and even the biological materials to see what they can learn about it and put the data back into the public domain. Sort of like the Cancer Genome Atlas Project, but even bigger and broader and with lots and lots of diseases and phenotypes. The reason I want to do that, and many people want to do it, is that we spend most of our time trying to build those groups of patients or individuals, not even like this would be healthy and people with disease and without disease, that we would do in this very large cohort. We spend so much of our time, it's incredibly inefficient, and progress would move forward so much faster. And I would put up tenfold faster if we had this. We've been talking about this for years. Francis Collins has been interested in it. Other leaders have been interested in it. We haven't had the will or the money to do it. Other countries are doing some things like this, but I'm not sure that the data are going to be freely available for everybody to use. So in some sense, it's not going to be useful unless it is broadly disseminated and without everybody worrying about whether they're going to get credit. This would be the ultimate thing like the Human Genome Project, but it's a lot more than genomics. So that's what I'd like to do. I don't even know where to start with how much that would cost, but it would take a lot of will and long-term thinking. This is a 10-year project to collect that, probably.
Definitely. Well, thank you so much for dreaming big with us today. And I think these big open data kinds of projects, you're absolutely right, could advance science so much, but everyone has to be willing to contribute and everyone has to be willing to play by the rules there. So wonderful project to propose for us and for us to think about moving towards in the future. And I think for all of our listeners out there who might be on their own scientific journey, it's really helpful to get advice, something that could help them along their own path. So Rick, is there a piece of advice that you can share with our listeners today that was really helpful for you?
Yeah, so this is a pretty personal one in that I mentioned I got into science late in college, in my third year, and I loved doing it, but I really didn't have a whole lot of confidence in myself about doing it because it still seemed like something that seemed unreachable and way, way out there, and you had to be a genius, and I'm smart, but I'm not a genius. I knew that. And so I had two professors in undergraduate school and one in graduate school who, as I was going along, and especially in undergraduate, encouraged me not, oh, you should go to graduate school, but here's one of your options. And they thought that I would be successful at it. I was not beating myself up. I just didn't know. I had not seen this. I had not understood how this would work. And their encouragement and that advice, and the advice was work hard. And clearly that was one of the things. I got to graduate school scared to death at Berkeley. Here I was coming from Alabama, assuming that I didn't have as good an education as anybody. It turned out I had a great background in very rigorous biochemistry from outstanding teachers and got lots of research experience. But I didn't know that. And so I still was quaking. I thought I was going to fail. I really had no idea whether I could do it. And I went through in my first year, I'm not going to mention his name, but there was a famous professor there at Berkeley who was quite scary. He was just really tough, but an outstanding teacher, an outstanding scientist. Took our first year that just about killed us. And I did really well in it. And he took me aside and told me that he thought I was really a good scientist, particularly in the laboratory. It was a laboratory. And honestly, until then, I had no idea whether I was good enough to do it. So it made me realize, okay, just stick with it, do it. I still had to feel like I had to work hard. But that advice was very, very helpful to me.
Definitely. And I think it's so reassuring for our listeners to hear that even someone as successful as you had these moments of fear, doubt, second guessing yourself.
I still do.
Yeah. Every day it happens. So, Rick, can you share any other last message with our listeners now coming from you personally?
Several things, and some of them I may have alluded to. I think the team science and collaborating is just absolutely don't feel like you have to do it all by yourself. Even if people tell you, yes, of course you have to prove yourself in a way, but get as much from other people as you can and share and collaborate. I think you can be competitive, but you do not have to lose your humanity. And you certainly see that happen. Not all the time, and I don't think anybody should ever stereotype all scientists as being one type or another. There's everything in between as well. But I take a lot of pride in trying to demonstrate that you can be a good human being and be competitive and successful, that they're not exclusive. I want to go back and emphasize again this education outreach program that is run by one of our faculty, Dr. Neil Lamb here. We put enormous resources into this. And again, I'm basing this on what I learned from Bruce Alberts almost 30 years ago. And one of the parts of that is that it's critical that we do service as scientists. And I think all of us owe this to the public. And some of my scientific friends at high-powered places think it's just fluffy and not important. I think it should be part and parcel of both our training and what we do on a weekly basis. We owe it to the public. They're the ones helping to pay for this. And that means lay people, legislators, people of different professions, and also K through 12 are actually what we refer to it as cradle to grave at Hudson Alpha. It's very early at every level, letting people know what we do, how it affects everyday lives. And there are ways to teach and describe this that don't have to be intimidating or showing off. There's everyday language. Neil Lamb happens to be the best person I've ever known in the world that can do that, but explaining why this is so important. And additional advice, I think that's something that I hope all young scientists, and actually ones my age and older and younger, would do. A couple of other things. There's not a single pathway to success in the careers. When I was at Stanford, we spent a lot of time with our students and trainees talking about, I guess we kind of used to call them alternative career pathways, but in fact, they're not necessarily alternative to academic professorships because maybe they're more common. I think being able to get training at whatever level, including high level in science, is extremely valuable. I would much rather have our leadership, our politicians, our people at various levels in companies, et cetera, to have scientific knowledge. And really, it's the scientific method, using evidence to make decisions as opposed to emotion. That's why it's a significant part of what we do here and a major part on the education side, but also teaching people who are getting this training that you don't have only one pathway to go. One that we always give students, I guess, even though I wasn't trained this way myself, biology has really become an information science. It's huge amounts of data, not just genomics, but the whole field is very data driven and lots of fascinating discoveries waiting to be found. And so learn how to be quantitative early on. And I'm so impressed with how the young folks, especially here, that's just almost like second nature to them now because they started it early. I think that's really, really important to learn how to be quantitative, computational, statistical, analyze, handle, interpret large data sets. And then finally, one more. I know many of my colleagues believe this as well. It's even more important now than it was when I was in graduate school. Learning a bunch of facts is not that helpful. For one thing, there are way, way too many facts. Then even in the late 70s, mid to late 70s, the amount of information seemed really large. It seems tiny compared to what you have to know now. And this is one reason why I didn't like biology in high school and science in high school, because all they taught us was a bunch of facts. It was very poorly taught. So what is more important, and maybe this is obvious, but it's hard to teach this, and luckily some young folks are getting this, is that you learn how to think. I mean, it sounds obvious, but you learn how to think, the process of taking in information and then what you do with it, because that information is going to change practically every day. So learning that and to learn how to think and to be adaptable. And that's especially important where the amount of knowledge has just skyrocketed and it's going to keep going that way. So I think it's learning tools, and this is where the quantitative stuff comes in too, learning tools about how to find information that you need to know, and then how you get what you want from that. How do you learn how to ask questions about that information as opposed to just taking a bunch of facts and doing something rote?
Well, Rick, you have left us a lot to think about, so many important points and pieces of advice that you just shared with us now. And thank you so much for your time today. Can you tell our listeners, though, if they want to learn more about you and your work, where should they go?
So go to our website, HudsonAlpha.org, or my email address is rmyers@HudsonAlpha.org. I will spell that RMYERS at Hudson, HUDSON, Alpha, ALPHA.org.
Perfect. Well, listeners, definitely reach out if you have any questions. Check out the website to learn more about what they're up to at Hudson Alpha. And Rick, thank you so much for sharing a piece of your own life and story with us today.
Thank you so much, Marie.
Well, this was a pleasure and wonderful to have you with us as well, listeners. We'll see you next time on another episode of People Behind the Science. Your voyage to explore the lives of today's exceptional scientists has just begun. You can find everything we talked about today, including our guests' favorite books, biographies, photos, and more, when you visit us at www.peoplebehindthescience.com. I look forward to chatting with you next time on People Behind the Science.